Welcome to another webcast in our series on antimicrobial peptides. I'm very pleased to be joined by Steven Theg. He is Professor of Plant Biology at the University of California, Davis. Professor Theg, thank you so much for being here today.
Sure.
We will be discussing a paper you published in March 2021, in which you talk about how the mechanism of TAT complex is poorly understood. Your research has been able to clear up some of the mysteries surrounding it. Could you tell us more about what you found and the significance of your findings?
Sure, I'd love to. The TAT transporter is a mechanism for bringing proteins across membranes. They're present in plant chloroplast membranes. They're present in bacteria, in archaebacteria, all the kingdoms of life uses this. But what seems to be unusual about this particular transport mechanism is that it doesn't use a proteinaceous pore to get proteins across the membrane. So proteins are pretty big, and lots of times they have to be unfolded and are threaded through proteinaceous pores. The TAT transporter, we think, causes the membrane just to break down. That is, the bilayer breaks down and makes what's called toroidal pores. These are transient small openings in the membrane, and then with the right circumstances, proteins can cross through these toroidal pores. So that's what this paper was trying to demonstrate. We were showing, what we looked at was the capacitance or the ion leakage across the membrane while we transported a protein on the TAT pathway. What we found is that we didn't see any leakage at all, no leakage that was above the limit of detection for the technique that we were using. Then we compared that to protein transport on what's called the SEC pathway. The SEC pathway does make use of a regular proteinaceous pore. And when we transported proteins on the SEC pathway, we did see some ion leakage across the membrane. So we were contrasting these two situations, I think, which points to, the way that it doesn't form these pores and cause leakage is more indicative of these toroidal pore mechanisms, these membrane destabilization mechanisms, as opposed to a proteinaceous pore.
And you write in your paper that the TAT complex might be the first known instance of toroidal pores being exploited by a general protein translocation system.
Correct.
And can you explain in further detail what you mean by this and how it affects the ability of the TAT complex to do its job?
There is, as far as I know, one example of a protein that goes across the plasma membrane it's got a dedicated transport mechanism, and there's a dedicated transporter with it. And it might be that this uses a toroidal pore mechanism, but it's a dedicated system. The TAT transporter, on the other hand, is quite general. You need a signal sequence on the front of it, and then you can have a passenger protein of any shape or size that you like. Now, that's an exaggeration, probably not every shape, but for instance, in E. coli, there are 32 different substrates that go on the TAT pathway. So it's a general transporter.
In what way do your findings help us to potentially develop new antibiotics?
Well, the TAT pathway is found in all walks of life. Part of the microbiome in the human gut is using TAT transporters. So probably for human health, there's a reason to care about this, that these transporters are required for the organisms that populate the gut. And so dysbiosis is a word that's used for when the microbiome goes sort of out of kilter and causes not necessarily diseases, but affects human health. Certainly, if we can find antibiotics that disrupt this kind of thing, we have a chance of working on this problem of dysbiosis. Now, we haven't found the antibiotics yet, but let me tell you the connection to the antimicrobial peptides, if I can. The mechanism, one of the mechanisms that these antimicrobial peptides utilize is this toroidal pore crossing the membrane. That is, it is thought that these antimicrobial peptides collect on the surface of the membrane. They cause the membrane to thin to the point that the bilayer breaks down. When the bilayer breaks down, you have these toroidal pores that form, and if the peptide's in the right place, it can slip across the membrane. This happens completely without a proteinaceous component. That is, the peptides go across the membranes all by themselves. Now, when we first started to think about this mechanism for the TAT machinery, we postulated that maybe the antimicrobial peptides, with their ability to thin the membrane, could get us sort of a head start on getting TAT transport across the membrane. And so we put them in with chloroplast membranes and asked to see if that would stimulate the TAT transporter, and it did. So these antimicrobial peptides, we used 4 different peptides that don't look anything like each other on the primary structure, but they all are antimicrobials. They all probably have this particular mechanism, and they all caused protein transport on the TAT pathway to be stimulated. We were excited by that. It was a prediction that came true. That's, so not proof positive, but, it's a prediction we made and it came true. So.
What do you think we can improve on further with more research?
Well, I would like to get the final proof for this kind of a mechanism. We're getting there by sort of coincidental things, like the prediction I just described to you came true. If we stabilize the membrane, stabilize the bilayer, that inhibits the TAT pathway. So things like that, but we get there slowly by a million different coincidental things that come true that after we make a prediction. That's all good, but we are yet to do the definitive experiment, which I think will be to show increased lipid transport from one side of the membrane to the other. That's a prediction that has to be true if you have this mechanism. So we've been trying to do that experiment for a long time. It's not an easy experiment, so we're still working on that.
Professor Theg, a big thank you for speaking on this subject.
Sure, I love this topic. Yeah.
Thank you,
yeah.
and
okay.
all the very best for 2022. Thank you.
Okay, thank you, Alistair.